I'm very excited to be here, to be honest, and to talk to everyone who's here. And today we're gonna talk about three other different tests from the wellness map. And those are the whole exome sequencing, and then the onco map, and then the third thing is the Neptune, where we're gonna golden, where we're gonna discuss the NIPT test that is specifically for the mother and the baby. However, we'll start now with the whole exome sequencing part. And for the WES map is done by blood. Let me just explain briefly what is whole exome. I'm pretty sure that everyone here is already familiar with it, but just for the sake for explanation. So the whole exome sequencing, basically we have the DNA, we have intronic regions, we have exonic regions, and this test is specifically for sequencing every single exonic area that's there in the DNA. So we're looking at the whole exome. We're not looking at certain specific genes. We're looking at the full picture without the intronic regions, which are not till now not known what they are coding for specifically. However, the whole exome part is a coding region, meaning that those parts of the genes are translated into proteins that have a function or a specific function in our body. So the first thing that we're going to see in front of us is the details of the patients, the test ordered by, sample details, etc. Then after that, we see a summary of what we've done and what we found as well. So we can see, first of all, on the right-hand side here, the sample quality and the data quality. And if we have the sample quality as passed and the data quality as passed, this means that our whole exome sequencing is reliable, or the sample is reliable, because we had enough DNA that we were able to sequence using our next generation sequencing. Then we have on the left-hand side, we have the test information. So what is the exome sequencing that we've done? And here we can see that we've done the whole exome sequencing along with the CNV, which is the copy number variation, which we're going to explain in a minute. Then we have here clinical information, and clinical information is very, very important. So for physicians that are here that are going to order this test, it is very important for the whole exome sequencing to have a clinical relevance, meaning that I've seen a patient, I see a condition with that patient, and I'm ordering the whole exome sequencing because of that. It could be family history. It could be a physical condition that when I observed when I was seeing my patient. It could be multiple factors, both factors together. So in this sample report that we see in front of us, we see a 2-year-old female with global development delay and hydrocephalus, meaning that the physician or the neurologist that was looking at this, that was dealing with this infant, they've seen that there is fluid around their brain. So in here, we see the clinical picture. Then it says that it has no family history, or there is unknown family history, meaning that both of the parents didn't have any issue growing up, or there isn't anyone in their family that has the same condition. And this, of course, gives a full picture for the physician when they are reading this report, to understand the history and understand where they are coming from, so they can put the right plan to treat that patient. Okay, then in the middle, we see the most important part, which is SNV positive, and SNV stands for single nucleotide variant, meaning that, and then we have CNV negative. CNV is copy number variation. And the difference between SNV and CNV is that SNV, when we all of us know that when we see the DNA, it's made of nucleotides. So it has A, C, and different other nucleotides, T, etc. Okay, so if we say that this is an SNV positive, it means that one single letter has changed in that DNA, causing the whatever we're seeing in that patient. So SNV is single nucleotide. From its name, you can understand what's happening on the DNA level. And then CNV is copy number variation. And when we say copy number variation, it is wider than SNV, meaning it is longer than SNV. We could see deletions, we could see duplications, meaning that, for example, if I have a piece of DNA that's ACCT, for example, I see that, of course, but the copy number variation is bigger than the example I'm giving. But I can see a repetition of the same nucleotides, right, one after the other again. So when we see that, that's duplication. I'm hearing an echo. I don't know. Someone does not have the mic muted. Okay, I'll continue. And then, so what I was saying is that we see duplication or we see deletion. That part of the DNA as a whole could be deleted from the patient's DNA. So that's basically the difference between SNV and CNV. SNV has specific variants that we're going to see, and then CNV, it could affect the whole chromosome because, again, it is longer, and when there is an alteration in the DNA that is longer, it has an effect on the whole chromosome. Okay. Then after we see the first part, which is a summary of the whole report, we can see in front of us the exact SNV that we've detected. So in here, for it, that's a sample report again. That's not something that we're gonna see with every single patient, but we see here the gene ID. Okay, then we see the variant, which variant exactly is present in that sample. And then we see the zygosity. So with zygosity, we mean whether it is homozygous or heterozygous. And by homozygous or heterozygous is that the baby or the patient has inherited it from both their parents, meaning that both his or her parents have they are a carrier of that specific gene. And then we see the name of the disorder. Okay, and then we see the inheritance, and in inheritance we can see multiple different results. For example, here we see autosomal recessive. However, we can see autosomal dominant, we can see X-linked, and we can see mitochondrial if we're sequencing the mitochondria as well. So what do we mean by that? Autosomal recessive is basically that both what I've just explained before, that both parents has it as a carrier and they do not have a disease. Autosomal dominant means that even if the baby has inherited it, or the person, again, it's not, the whole exome is not only for babies, it's for everyone, or the person has inherited it from their mother or their father, they have a dominant effect, meaning when the alteration is there in the DNA, it will most probably project a disease that is visible in the person. Then we have X-linked. X-linked meaning that it is related to the X chromosome. And specifically, the X-linked ones have a difference in when they show in the patient whether they are a female or a male, because again it is linked in the specific X chromosome, which is as we all know, is two X's in the females and XY in the males. So that's why it has a difference when when it is a female or a male. Then we have in the X-linked has two different variation, the X-linked recessive and then the X-linked dominant. And also it's the same thing, recessive meaning it could be a carrier, but dominant means that it will project a specific disease that we can see in the patient. Then after that, we have the variant classification. And the variant classification, it is a global thing that is known in the American Genetic Society. We know whether it is pathogenic, likely pathogenic, VUS, or likely benign or benign. So in the variant classification, we have five different results that we could see when we're seeing those kinds of reports. So pathogenic means that the patient has a disease or it is classified as a disease. Then the likely pathogenic, it could be a disease. VUS means that it is a variant of uncertain significance, meaning that we cannot say whether this specific variant could cause a disease or not. So it is still unknown in the scientific world. Then we have likely benign. Likely benign means that it most probably does not cause any diseases. And then benign, that it is for sure doesn't have any relation to the disease that we're seeing or whatever disease that it could be. Okay, I want to stop here for a second. I want to make sure that this part is very clear because it is basically the most important part in the report. If you have any question about this part, please let me know. Okay, I think we do not have any questions, so I'll move on. Then after that, we see the detected CNV variants. As we said before, the CNV is the copy number variation. In that specific sample report, there isn't any CNV that is detected, but if it is detected in any other patient, it will be written in this part. Okay, after that, we have the medical interpretation, which is very, very important. The whole exome sequencing could be a very challenging report to understand, but a wellness map have the medical interpretation integrated into the report, so it guides the physician to understand what is the condition that this patient might have and how they can understand it more. I'm not going to go through it specifically because this one is very specific for each sample report. But again, in this part, you're going to see all the details that could be related to the medical condition that this person might have, and then it has a recommendation in the end where it says that, for example, here that this patient has a severe condition of hydrocephalus, okay, and that further clinical indication needs to be distinguished whether it is the type which is A3 or not. Okay, so and then after that we see the genetic interpretation. Again, this part is also a summary of what we is details of the summary that we've seen before. So in the genetic interpretation, we see the gene ID, the transcript ID as well. We see the location of which chromosome it is on. Okay, we see the coverage. Okay, we see the zygosity, whether it is homozygous or heterozygous, and we see the inheritance pattern, which is we've seen before, which is autosomal recessive. And then after that, we see the disease here. What are the type of diseases that could be related to this specific SMV? Then after that, we see the classification, and the classification, it means that there are multiple classification words in the genetic societies. So we have the ClinVar, and we have Franklin, and we have Varsum. So those three are basically like databases that are out there that are available as well. We can always take the gene ID or the location and or the transcript and put it in these specific databases to understand more about those genes and that variant that we've seen in this patient. And then so that's the classification database. And then in the classification, we see whether it is pathogenic or likely pathogenic. Then after that, we have also a summary that is very detailed about the gene itself. So it says here, for example, that we have this gene that is located on the chromosome or exon 16 out of the 22 chromosomes that we have. Okay, and that variant, it is classified as pathogenic, which is also what we've seen in the summary. And this was compared to Franklin and Varsam database. So also to know which database that Wellness Map has used to produce this report. And then we see which criteria it follows. And the criteria here is the American College of Medical Genetics. Okay, so it was so this is what we've seen when we or what we've discovered when we were seeing whether it is pathogenic, likely pathogenic, VUS, likely benign or benign. And that's it. And then it tells you what are the IDs that we can use to put in the database and see the variant with the details and the publications that have been published that are related to the whole, that are related to that SNV specifically. Okay. Then after that, we have the recommendation. And in the recommendation is basically what are so those tests are usually reviewed by a geneticist because the whole exome sequencing is usually related to diseases. So it's not only screening or wellness type of a test. It is most probably related to a clinical picture that I've explained in the beginning. And the geneticist starts putting those recommendations in the recommendation section here. So we can see here that it has to be followed up with the referring physician that it needs a genetic counseling, the specific case. And then it tells you that those variants could be found in the parents. So the parents have to do whole exome sequencing as well to understand whether they have this variant or not, because this could affect their offspring as well. So it is very important for the family planning. And then it tells you here whether it has that even though that CNV is not detected, but CNV with NGS is different than CNV with other techniques. And maybe it is important to do that the CNV on its own using microarray, for example. And that's it in the recommendation. Then whether there is secondary finding or not. For example, the sample report doesn't have a secondary finding. However, when we are doing whole exome sequencing, it is sometimes the genes that we're looking at that are specific to this type of disease. We can see other secondary findings that are not specific to the disease that we are sequencing for. So sometimes it is, we can find the secondary finding and it will be put in that section. Then after that, we have the citations, like the databases that were used. It could be also the publications that the geneticists looked at when they were looking at this report, etc. So that's it for the wellness for the whole exome sequencing. Please let me know if you have any question.
Questions?
Thank you, Sandra. I have a question.
Yes,
So,
please.
for me, for example, I'm a family doctor. Would this be something that I request as a family doctor, or and usually it's requested by people who are more specialized?
Yeah, of course, a family doctor would be a perfect person to prescribe this test because you see a lot of patients and you see a lot of kids, you see a lot of parents, etc., and individuals that
Mm,
might have a condition. When you're checking the patient, you can, uh, by your knowledge, know that it is most probably a genetic disease, or
mm,
that there isn't anything that's for example, the parents do not have anything that's physical, but you see something different in their child. So the whole exome sequencing is something that is very important to prescribe as a family doctor, especially when you see a type of hereditary disease that is physical on the patient, and you can and you do not know, or the family does not have any history in that. So the whole exome is very recommended for such patients, for example.
Thank you. Thank you so much.
You're welcome.
Question, Sandra. Regarding prenatal and postnatal whole exome sequencing, what you recommended and what is the differences between both?
So, when we're talking about whole exome sequencing, we're talking specifically about the test that we do as patients in general, not specifically prenatal or postnatal. When we do a test for, let's say, embryos, we're going more for the PGT kind of a test, not whole exome sequencing specifically. And when we're doing postnatal, it is very important because in our communities there are a lot of marriages that happens from the same family. So the postnatal whole exome is very, very important because it would detect if there are any hereditary diseases that are there. However, an IPT test in that specific let's say process is important in the middle, and it shouldn't be prenatal or postnatal. We have to do the NIPT to make sure that the embryo or the fetus does not have any issue before giving birth.
Is it enough to do single whole exome sequencing, or you prefer as a doctor to do the trio exome?
No, three whole exome, of course, is very important. So it's very important to do, like, for example, if we see this specific case in front of us, the whole exome sequencing was done for the 2-year-old female specifically. It was not done for the parents. And then the recommendation came that it has to be done with the parents because genetics is basically hereditary. So it's the mother, the father, and then what they do, the DNA that they get is basically a mix of both. So it is very important when we have the postnatal, we have to do the for the infant and both parents, so we can link the diseases together if there are any, of course.